There we go. We're recording. We have our culture tubes, each with 2 mLs of luria broth, plus the ampicillin, plus the X-gal. Hold on a second. There is no X-gal in here, but there is ampicillin. This is luria broth plus ampicillin. Where's the X-gal, you might ask? The X-gal is also with ampicillin on these plates. So these are the plates I mentioned. They're a little foggy from condensation. These plates, there we go. See the blue dots, guys? Somebody say yes.
Yeah, we were using the blue dots.
Thank you, Adrina. Can you see me behind them? Wong, wong.
Yeah, barely.
Barely. I'm here, I promise. Do you want the blue dots?
No.
You want the white colonies. Why?
Because they actually have the gene inserted into them.
Exactly. They have the cDNA inserted into the LAC-Z gene. We do not want those blue colonies. So let's see if we can get you some white colonies and grow them in this luria broth with ampicillin, the food with ampicillin. Again, this has food with ampicillin that kills bacteria. The plate has food, ampicillin, and X-gal. The X-gal turns blue in the presence of the beta-galactosidase gene. Okay, here we go. Now these plates are, there's a lot of blue colonies here. Wow, a lot of blue. All right, so I'm holding them up to my light up here, and I can have a better little view of it. If we were in the classroom, I could probably see, you guys could probably see better, but I'm gonna go for it here. All right, here we go. I got you, I got you, I got you. I can get 6 from here. All right, so these are sterile sticks. They've been autoclaved. They're wooden sticks. Pinky, remember that? Take out the sterile stick. Pinky goes right back. See that? I never put that down. The stick is sterile. That's the business end. That's sterile. It's about to be not sterile. I find my target on my plate. Now my target is a white colony, as we just mentioned. That should have an insert. I gotta find a good one. And by golly, I think I found one. Here we go. I'm gonna do my best to get the picture of this in the video. So I touched it and I closed this plate. Now I clamshelled this plate. I never fully opened it because I don't want to breathe onto it. I don't want to infect it with anything from my mouth. So I'm going to go T1. It's T1. Pinky. See, pinky. Bacteria on the tip of the stick. Drop them in. Stir. I've stirred my cells in the luria broth with the ampicillin, and I get the cells off, and I'm really jamming them in there. And the cells are fine. They have peptidoglycan cell walls, bacteria do. E. coli are gram-negative, peptidoglycan-rich cell walls. They're fine if I can smack them around. They don't care. They're happy to be in food and liquid. All right, so I jingle that off. I put the top back on. I smush the cap on. I'll double check later for that. But this is done. Crack. I'm done with this stick. I don't want to use this stick again. That goes in the garbage. Let's do number 2. T2. Stick. Back on. Plate. I have a light up here. I have a good colony. I see one. All right, T2, here we go. Boop. I just booped it. I'm booping it. I just boop it. I don't smush it. I just booped it. The boop is important because I don't want to smush the agar. The cells are right there on the wood. Take my T2. Take my cells and put it in T2. There you go, T2. That's all you. That's how we do for T2. Spin it up, spin it around, jingle it. Look, I never put the cap down. It's right there. I'm like David Blaine. Sleight of hand. Jingle it around. Get those cells off. They're happy to be there. Get rid of this stick. You're dead to me, stick. I put the cap back on. I smush the cap back on. I'll talk about that in a minute. All right, T2, you are done. Let's do T3. Another stick. Never put the cap down. Now, if you guys were doing this, I'd tell you to find the target first. I've done this a bunch of times, so I'm pretty happy with the targets. I saw them before. There's my target. I always put it at 12 o'clock. Open the plate, hold my breath a little bit. Close the plate. The cells are on the end of that stick. T3. Cap comes off. Cells go in. I wiggle it just a little bit. Get those cells all up in that food, that luria broth.
Mr. Jones.
Yeah.
Have there actually been any major consequences that have occurred since if someone actually breathes onto the bacteria?
Onto the plate?
Yeah.
Destroy the stick. I wouldn't really know. Although, Adrita, I'll tell you this much. These plates, once I use them once, after a couple days, I will get fungal puffs on them. And I'll get weird bacterial colonies growing. Because little bacteria and fungal spores from our mouth, and we all have it. We have yeast in our mouth. We have bacteria in our mouth. We eject it when we talk. That's why we wear masks. And the bacteria can fly out and land on the plate. So it's really hard to not do. Mm-hmm. And that's why, Adrita, I clamshell it. Now, if I was in class, I do something called booping. I boop it from behind. I turn the plate around, and I come around like this and boop it from behind. A little hard to do on camera, so I didn't boop it from behind. I'm booping it from above. Let's boop it. So yeah, it's hard to avoid. But you know what? You just gave me an idea. If we're wearing masks, that could really help, couldn't it? That could really help. All right, where am I at? I'm at T4, right, guys? Anybody?
Yeah, T4.
Thank you. Good. So T4, let me find a good one for you. T4, good one. Oh, I got a nice one right in the middle. That bad boy right there. Here we go. Here we go, T4. That's a big one. Now, a big colony may mean a small insert. Look at that. I got it all. Can you see it? You can't. It's invisible. Nor can I. We have to have faith. Cap comes off. Cells go in. Wiggle and jiggle. Wiggle and jiggle. Get those cells mixed up with the food. That's good resolution. It's not bad. All right. Get rid of that stick. Cap goes on. T4. All right, T5. Let's look alive. Cap goes back on. T5. Wow. Is there? Oh, I think I got a good one. T. There's a lot of whites with blues. I'm avoiding those. I see a lot of whites with blues. Ooh, I got one for you right up there. Here we go. Top of the plate. All right, here we go, T5. Got it. 5. Cap. Look at that resolution. That's amazing. Got to give it to you, Apple. I'm not an Apple guy, but that's a good camera. iPhones have good cameras. Okay, good. That should be good. Smush it. Okay. Last but not least, tabla 6. All right, table 6. Let's see. I'm looking for a good one. I want to find you guys a good one. Oh, I need some natural light. I really should have done this before I picked the stick up. You know what? I think I got one. It has a little bit of blue in it, but that's okay. Trust me on this. All right, it's mostly white. Here we go. Right there. Boom. Got you. All right, got you. Close it up. Table 6. Mix it up. Wow, I can see myself in the tube. That is trippy. All right, good. Table 6. Up and down a little bit, around and around. Cap goes back on. All right, I'm going to smush these caps on. Listen for it. Snap. 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 I already smushed it. I'm just double checking. Snap. There we go. Snap. Now, see, these caps are great because these are fluted caps. These are not like screw-on caps. These caps are really on tightly. It should work. It should work if it falls over. But the caps are great because they let air in and out, but not liquid. And that's important because we want the bacteria to get rid of what? What do all cells produce?
Carbon dioxide.
Exactly, right? They have to make some well, E. coli definitely makes CO2. Do all bacteria make CO2?
No.
I think even the chemosynthetic ones make CO2. The chemoautotrophic ones, because they generate their own food molecules using other electron donors. So I'm almost certain that every single cell on the planet will make some carbon dioxide. And yes, we have to get rid of it because guys, if you have excess CO2 in here, what happens? What does it form?
Bubbles?
Carbonic acid.
Carbonic acid. You got it. Carbonic acid. And what happens to the pH?
Down. It drops.
Down. And they're not happy, right? Cells like a regulated pH. So we have to get rid of the CO2. Furthermore, now E. coli, while it's anaerobic, would like some fresh air, I imagine. So basically, gas exchange. Gas exchange is important. And that lets excess gases leave and the pH to kind of stay constant. Because the CO2 will bubble out. Not all of it forms carbonic acid. There we go. So our caps are on tightly, and we're ready to bake these things in the Incu shaker. Let's bring the Incu. Here we go. Jonesy's Garage Incu shaker. Oh, you have. Let me do this. Maybe that's better. That's better, isn't it? Hold on, I'm gonna turn the light on. That should be better. There we go. Hello. So this is an Incu shaker. I have it spinning at 200 RPM. and at almost 37°C body temperature. So the body temperature is great because bacteria love almost 100 Fahrenheit, almost 40 centigrade. They don't love much higher than that. E. coli loves 37°C, loves 98.6 Fahrenheit. That's why it loves to grow inside of us, in our intestines and other places, unfortunately. But a lot of bacteria love human body temperature. Now, the shaking is interesting. I'm going to put these in. Think about the shaking. This is you guys. I'm going to put you guys in the back because you're up here at 12. Before I close it, though, this is from this morning. This is you guys. Does anybody see a difference yet?
One looks less cloudy.
Does one of them look cloudy?
Yeah.
A little bit, yeah.
Yeah.
Very true. That's only 5 hours ago. Not even. Right? So that's pretty good. That's less than 5 hours ago. That means we're getting growth. That makes Jonesy happy. So this is you guys in the back. All right. Almost dropped you there. Okay. Caps on, caps on. Close the hatch. And there they go. So why is the shaking important? 200 RPMs overnight.
Does it separate the cell bunch into so it has more surface area with the food so it grows faster?
That's exactly right. Yeah, it's not centrifugation. We're not spinning them. We're just vibrating them. So this is not centrifugation. This is just vibrating it. But you're exactly right. Who said that? Was that Shraddha or Sia? Shreya?
Sure.
Shreya, I'm recognizing. So Shreya is right. The cells can get to food more easily if they have constant food flowing over them. The shaking is like 80% better growth. It's amazing. They love to be shaken. So think about it. This is your body temperature. This is bacteria. It stands to reason how bacteria would grow very well in our intestines, because our intestines are constantly doing peristalsis, right? Squeezing, whether we know it or not, smooth muscle. Your intestines are constantly 37°C, 98.6 Fahrenheit. Your intestines are great places to grow bacteria. That's a little gross, I know, but that's the truth. And that's what we're trying to kind of replicate intestines. And this is a proven method to do so. It's called the InQ shaker. All right. So we'll let those grow overnight. And we'll talk about tomorrow what we're going to do to them. How are we doing on time? We had 10 minutes. Nice.
Wait,
me-
Mr. Jones, did you that on for the entire night?
All night. Yeah, the whole night I'll let them grow. I'll let them shake all night. Take my gloves off. Adjust the lights. Take a couple questions. Do you guys have any questions on that overnight growing of bacteria for the video?
So do the caps of the tubes, the test tubes, do they have that kind of like Louis Pasteur curved neck? Like, can bacteria get in and out?
Oh yeah, very good. The Louis Pasteur curve neck, yeah, would be like that. Whoop. That's to prove that bacteria can't fall in. These don't have that. All these are. I know the ones you just saw were blue. I get that. This is a clear-ish one, but it's the same thing. These don't have that. These are simply open. This is a tester one. This is an open with a fluted top. That's all
So
that
would
it is.
bacteria hypothetically be able to get in?
Oh yeah, definitely. Once I open this, bacteria can fly right in. And
Oh wait, so what is the fluted cap referring to?
these little like, see if you notice this cap, it doesn't screw on, it smushes on. See, I'm gonna push. It smushes on. So the cap, it smushes because it lets air in and out, but it doesn't screw, because threads for a screw they'd keep liquid and air out. Threads would, because threads press against each other. This presses against itself, but there's flutes. There's little like there's little lines in here, and that allows. We could allow Millie in. Millie's not from outside Edison. Okay, whatever. Did you leave, Millie? I don't know. Anyway.
Sorry, my Wi-Fi keeps kicking me out.
No problem, no problem. It's okay. Yeah, so the flutes allow gases to exchange. They're like little bumps that allow gases to go up and down.
Okay, thank you.
You're welcome.
Wait, so would the bacteria hypothetically be able to go in through the flute with the cap closed?
No, I didn't show you this, but if fluid will not leave this. If I hold it upside down, yeah, I'll quickly show you. I have some. This is a dummy tube anyway. This thing is dirty. If I squirt some, if I put some Simple Green in here, just as an example, let's pretend this is bacterial fluid. If I do this right, the fluid shouldn't come out. Let's check. It's over my computer, yeah. So fluid holds in there.
What about like airborne bacteria?
Yeah, bacteria could never fit inside this.
Oh, okay.
They couldn't get past the flutes. But oxygen and CO2 can. Nitrogen gas, noble gases, little small, diatomic gases and triatomic compounds could get in and out. Small ones, maybe even alcohol, maybe ethanol could probably get out if it evaporates out. So like,
Okay.
you're welcome. So these would not be good to make like beer or ferment any liquid or anything like that. I can even shake it and you can see bubbles forming. But if I squeeze it, see, I can squeeze this and the air comes out of it as I squeeze it. Excellent question. So I have 25 left for 2 more classes. That should about do it. 6 times 2. I'm at about 12 each. Yep. So that should be good. All right, I'm going to stop recording just to keep this a little short.